The endoplasmic reticulum, or the ER, is a critical cellular organelle involved in protein synthesis, folding, and quality control. It plays a vital role in ensuring that newly synthesized protein are properly folded before they are transported to their functional location within the cell or secreted outside the cell. They are also serve as a hub for lipid synthesis and calcium storage. But now, what will happen if an ER storage malfunction? So, welcome everyone. I am Shirley and Makiji. Today we will be discussing ER storage diseases. So, endoplasmic reticulum storage disease, or ERSDs, are a group of genetic disorders characterized by accumulation of misfolded and misunfolded proteins in the endoplasmic reticulum, or the ER, a cellular organelle involved in protein synthesis and folding. The term endoplasmic reticulum storage disease (ERSD) was introduced into the medical literature after discovering that similar to alpha-1 antitrypsin (AAT) due to the genetic mutation, another acute phase protein, fibrinogen, and could undergo misfolding within an endoplasmic reticulum (ER) instead of being transported into the blood. The two related diseases were designated alpha-antitrypsin deficiency, the AATD, and the hereditary hypofibrinogenemia with hepatic storage, the HHHS, together with other intercellularly or extracellularly misfolding proteins. ERSD are classified as a conformation disease and included together with the mitochondrial, lysosomal, and peroxisomal disease into the group of pathologies of cells, organelles, and into the metabolic disorders and pathologies of the liver. The accumulation of misfolded proteins triggers a cellular stress response known as the unfolded protein response, the UPR. The UPR now is a complex signaling pathway that aims to restore ER homeostasis by increasing the production of chaperone proteins, which assist the protein's folding by reducing the synthesis of new proteins to alleviate the protein folding burden. If the UPR fails to restore normal protein folding, the misfolded proteins can aggregate and form insoluble structures, causing ER stress and cellular dysfunction. This can lead to various diseases manifestation depending on the specific ERSD. Now, ERSDs can affect different organ and systems in the body depending on the tissue-specific expression of the mutated gene. For example, some ERSDs primarily impact the liver, leading the liver disease, while other predominantly affect the nervous system, resulting in neurological disorders. For examples of ERSDs, including alpha-1 antitrypsin deficiency, as I said before, the AATD, as we discussed earlier, as well as diseases like cystic fibrosis, familial hypercholesterolemia, and various forms of hereditary neuropathies. So we have example here of a figure that is an ERSD in under a microscope. But today, for an endoplasmic reticulum storage disease, we or I have chosen a disease that is the AATD, the alpha-1 antitrypsin deficiency, the prototype of ERSD. So, alpha-1 antitrypsin AAT deficiency is a genetic condition which increases the risk of lung and or liver diseases. People with AAT deficiency have low levels of AAT protein in their blood and are more likely to get lung disease earlier in their life than someone with higher levels of AAT. The most common lung disease that people with AAT deficiency are likely to get is chronic obstructive pulmonary disease, or the COPD. So alpha-1 antitrypsin AAT is a major component of a alpha-1 globulins, is the major serum proteinase inhibitor, especially active against leukocytes elastase. AAT is also an acute phase reactant capable of raising its serum level up to quadruple the normal level under condition of clinical simulation. It is thought that the less than 1 in 10 people with AAT deficiency have been diagnosed. This means that most people with condition are unaware that they have or they could benefit from changes to their lifestyle or medical health, which could reduce the chances of getting lung diseases. Now, AAT protein is made up in a liver and releases into the blood. It enters the lungs from the blood, and its job is to protect the lung tissue from being damaged. It protects the lungs from another protein called neutrophil elastase, which is released by white blood cells. The job of neutrophil elastase is to destroy damaged cells and bacteria. AAT stops it from attacking healthy lung cells. In individuals with AATD, a mutation in the SERPINA1 gene leads to the production of an abnormal form of alpha-1 antitrypsin AAT. So, a protein primarily produced in the liver, this mutation causes an AAT protein to misfold, preventing it from being properly released into the bloodstream. The misfolded AAT protein gets trapped within the liver cells, specifically in the ER, or the endoplasmic reticulum, which is responsible for protein synthesis and folding. The accumulation of misfolded AAT in the ER triggers a stress response known as the unfolded protein response, the UPR. The UPR now attempts to restore the normal protein folding and preventing cell damage. However, in the case of AATD, and the UPR is unable to, or uh, to effectively address the misfolded AAT protein, leading to further accumulation and causing the liver cells to become overwhelmed. This accumulation now of misfolded AAT in liver cells can lead to liver diseases such as the cirrhosis or the hepatitis. However, the most significant impact of AATD is observed in the lungs. Without sufficient function AAT, the lungs are more susceptible to damage caused by an enzyme called the neutrophil elastase, which can break down lung tissue. The imbalance between neutrophil elastase and the AAT can result in chronic inflammation, tissue destruction, and the development of lung conditions such as the chronic obstructive pulmonary disease, the COPD, and emphysema. emphysema. To manage AATD, various treatments options are available. This includes the augmentation therapy, which involves infusing a purified AAT protein into the bloodstream to increase levels in the lungs, and other supportive measure of to manage symptoms and prevent further lung damage. Understanding the mechanism of AATD has paved the way for ongoing research into protein therapies, including gene therapy and small molecule drugs that aim to enhance the production, folding, and secretion of functional AAT. So here we have an illustration on how a person without an AATD uh, circulate the uh, blood. So without the AAT, the AAT is produced in the liver and travels to the lungs, protects them from neutrophil elastase. So I've said before. So in here, the AAT will travel to the liver with protection against from the neutrophil. Now, however, but in a case with a person with a severe AATD, the AAT does not get in the lungs. This means that they do not have protective coating and are damaged by neutrophil elastase. Now, AAT is produced in the liver but cannot leave the liver. AAT stays in the liver and can cause liver damage. Now, this disease during childhood, individuals with AATD typically do not show any symptoms. However, as they reach in their 20s or in their 50s, the symptoms may start to manifest. The severity of the symptoms can vary among individuals depending on the specific mutation and other factors. The most common noticeable impact of ADD is on the lungs. Without a sufficient functional AAT, the lungs are more susceptible to damage caused by enzymes, particularly the neutrophil elastase, which can break down the lung tissue. This can lead to chronic obstructive pulmonary disease, the COPD, emphysema, or the severe case respiratory failure. Several different abnormalities in the SERPINA1 gene have been found that result in different amounts of AAT proteins being made. This means the condition can be severe in some people than others. Now, how can genes cause AAT deficiency? DNA is a code that makes up every part of our bodies. There are millions of tiny sections of this code, and they reach do different things. These small sections are called genes. We have two copies of almost all our DNA, one from our parents. Now, the two copies of parents' DNA. AAT deficiency is caused when there is an error abnormalities in the code in one of a gene. This gene is called SERPINA1. Now, the severe AAT deficiency is called recessive condition. This is because there need to be two abnormal copies of gene that cause the condition. So, in short, the AATD is caused through genetics. People who have one abnormal and one normal SERPINA gene are called carriers. They usually have an intermediate AAT deficiency. This means AAT levels are usually lower than the normal, but not low enough to cause serious risk of health problems. They can pass their one abnormal genes to their children, but they do not have the condition themselves. So, inherit. The children of two carriers can inherit the normal genes, one normal and one abnormal genes, and become carriers themselves, or two abnormal genes and have an AAT deficiency. Here we have seen this diagram on how can genes cause AAT deficiency. So to sum up, here is the pedigree of how parent pass through their child. First is the yellow. If the two are carrier, one can be the normal, two can be carrier, and AAT deficiency is one. So if a person is one carrier and AAT deficiency, the possibility of having deceased child is 2, and the rest can be a carrier. So here yellow means normal, the yellow blue means carrier, and blue means the AAT deficient. Side note, does having AAT deficiency means you have or we have the disease? Actually, not really. AAT deficiency is a genetic condition which increases your risk for developing lung and or liver disease. Although many people with AAT deficiency who do not smoke do not develop any disease and or related to the symptoms. So actually, it progresses over time. It's about a lifestyle of a person. However, it may have a higher risk of having lung diseases if you ever have an AAT deficiency. So, how common is AAT deficiency? Although AAT deficiency is thought of as a rare genetic condition, it is actually one of the most common inherited genetic condition in some part of the world, such as Europe. In some northern European countries, between 1 in 1,600 and in 1 in 2,000 people are thought to have an AAT deficiency. In some other regions, in Southeast Europe, AAT deficiency is less frequent. Recent studies estimated that there are about 120,000 people in Europe with severe AAT deficiency. The reason that people think it is rare is because disease associated with AAT deficiency are often not recognized or diagnosed. There are often a long delay before people are diagnosed with condition even after visiting a doctor with symptoms. One specific study found that diagnosis of AAT deficiency was made on average 7 years after the first symptoms appeared. So the reason that most people with lung diseases associated with AAT deficiency are either not properly diagnosed or are never diagnosed at all. It includes symptoms can be very different in some people. COPD or asthma symptoms can have many other causes. Liver diseases also can have another causes. There are many things that people with AAT deficiency can do to ensure that they have lower chance of getting lung diseases or to slow its progress. Most of these involve trying to avoid damaging the lungs as there will not be enough AAT to protect them. Not smoking, smoking is even more harmful to people with AAT deficiency as their lungs have less protection. Avoiding highly polluted areas if possible, avoiding exposure to fumes or inhaling toxic substances in the workplace, preventing bacteria and viral infection by avoiding people who are unwell and by being careful about personal hygiene and having prescribed immunization. So eating a balanced diet rich in protein and vitamins are one. Also, performing breathing exercises as recommended by your doctors. These are some ways to prevent uh, lung diseases with AAT deficiency. In conclusion, alpha-1 antitrypsin deficiency or the AATD serves as a prototype for understanding ER storage diseases. AATD is a genetic disorder characterized by production of abnormal alpha-1 antitrypsin AAT protein leading to its misfolding and accumulation in the liver cells. This triggers the unfolding protein response, UPR, but fails to effectively address the misfolding AAT protein resulting in liver diseases and lung damage. AATD highlights the critical role of protein folding and quality control mechanisms in maintaining cellular homeostasis. The accumulation of misfolded protein in an endoplasmic reticulum ER can lead to ER stress, cellular dysfunction, and the development of specific diseases manifesting. Studying AATD has provided valuable insights into the mechanisms underlying ERSTs, a group of genetic characterized by the accumulation of misfolding protein in the ER. These insights have paved the way for further research into understanding protein misfolding, ER stress, and the development of potential therapeutic strategies ongoing. Research aims to develop innovative approaches to manage ERSTs, such as pharmacological chaperones, gene therapy, and small molecular drugs that modulate the unfolding protein response UPR and proteostasis pathways. These advancements offer the hope for improved better outcomes for individuals affected by ERSTs, including AATD, by shedding light onto the development mechanism and broader implication of AATD within the context of ARSTs. We can deepen our understanding of these complex disorders and drive progress towards more effective diagnostic intervention and therapies. So that's all for today. I hope you have learned something, and here are my reference. Thank you.